Good morning, everyone. My name is Garcia-Baccino, and I will present our work entitled Detection of Unrecorded Environmental Challenges and Genetic Determinism of Resilience in Lambs. As you can see, this is joint work with Marie-Etancelin, Flavie Tortereau, Didier Marcon, Jean-Louis Weisbecker, and Andrés Legarra. Animals can be exposed to different types of challenges or disturbances, as for example nutritional changes, management and farm conditions, weather conditions, health challenges, among many others. These challenges or disturbances can have a negative impact both on productivity and welfare. So when we speak about resilience, we refer to the capacity of an animal to be minimally affected by disturbances or to rapidly return to the state pertained before exposure to an environmental challenge. Given the general characteristics of small ruminant production systems, animals are commonly exposed to heterogeneous and changeful conditions. That is to say, that there is little control of environmental factors compared to other livestock species in which production systems are more intensive. Consequently, it is necessary to select animals capable of maintaining their production performance or modifying it as less as possible under this heterogeneous environment they are exposed to. When an environmental challenge affects the whole population, it is a good opportunity to select for resilience, but usually under real productive conditions, challenge events are unrecorded or only partially recorded, and also from unknown source. In this sense, our objectives are first to present a simple and practical data-driven approach to detect environmental challenges. The idea is to estimate the probability that at a given date there was an unrecorded environmental challenge occurring, and to do so we use a mixture model of phenotypic variances. Second, to evaluate genetic determinism of resilience to these events using these probabilities as covariates in a reaction norm model. And finally, to illustrate the method using a real ovine dataset as an example. Basically, the idea is that the effect of environmental challenges on animal performance can be observed indirectly through changes in variability patterns in repeated records of performance over time. This can be seen in this figure. It is an example in which we can see that there is an increased variability for feed intake for days 3 and 8 as it is shown in blue, and this can be associated to disturbances. This was observed in many cases for different livestock species. For example, in 2016, Kolditz and Hein and Frigens and co-authors reported this kind of changes in variability patterns in small ruminants when exposed to thermal or nutritional challenges. And more recently, in 2020, New reported similar results for feed intake in pigs. In all cases, variation increased related to the occurrence of an environmental challenge. So, our method is as follows. First, we detect environmental challenges. In this sense, we can think of two kinds of days. On one hand, those stressful days that are those with high variance, and on the other hand, not stressful days that are those with low variance. We can detect them fitting a mixture model to raw data, and on output we can have 2 or maybe more components. The component related to larger day variance is associated to stress. And we can also estimate posterior component membership probabilities for each day. These are probabilities that a given day belongs to the first or the second component of the mixture. So then we can evaluate genetic determinism of resilience. These probabilities we estimated before can be used with 2 different purposes, either for clustering, let's say to differentiate between normal and stressful days, or directly as an environmental descriptor, continuous variable, to characterize the environmental gradient. And in this last case, we can include them, the probabilities, as a covariate in a reaction norm model. We can see here that probabilities close to 0 correspond to non-challenging conditions, while values of P, of the probability close to 1, are highly variable environmental conditions. We employed an ovine dataset with daily feed intake records as an example to illustrate the method. In the dataset there were 951 Roman male lambs, all with the same age, 12 weeks approximately. All the animals were phenotyped for feed intake, and the phenotypes were continuously recorded using automatic concentrate feeders at the INRAE Sapinière experimental farm. Every year, over an 8-year period, that is to say, going from 2009 to 2016, a cohort of animals was phenotyped during 8 weeks in winter, and on average, there were 119 lambs tested each year. All animals within each year were tested under the same environmental conditions. There were almost 52,000 daily feed intake records available and 5,114 animals in the pedigree. And here in the pictures you can see an automatic concentrate feeder used to record the data we worked with. So first, to detect environmental challenges, we assessed variation of daily feed intake using the coefficient of variation. We fitted a mixture of two Gaussian distributions to the logarithm of coefficient of variation, as you can see in the figure, where we can see the two components. One is in red and the other one is in green. And we were able to obtain posterior component membership probabilities for each day, as you can see there in the list. So, as you can see, for example, the probability of being a stressful day for day 1, that is the first line in the list, is 0.58 approximately. That is the probability of pertaining to the second component that, as you can see, is in green. We obtained posterior component membership probabilities for all the days, as you can see in this figure. We have one figure per year, and on the y-axis we have the probabilities that at a given date there was an unrecorded environmental challenge occurring, that is to say the probability of pertaining to the second component of the mixture. And we have the day on the y-axis. As you can see in red, we detected some days with probability of being stressful higher than 0.9. We checked the information recorded in the logbooks of the experimental farm, and we confirmed that there were environmental challenges recorded for some of those days that we found. Then, on a second stage, we evaluated genetic determinism of resilience. As I said before, we were able to associate each day to a probability of being a stressful day, and this enables a characterization of the daily environment understood as a combination of different variables to which all animals were exposed to. As you can see here in the figure, values of probability close to 0 correspond to non-challenging conditions, while values close to 1 are associated with challenging conditions. So the model we employed was a reaction norm model with P, the probabilities, as the environmental covariate. Here we can see the model, and where we have the daily feed intake record here, and here in the first term we have a combination of year and automatic concentrate feeder fixed effect. Then on the second term we have a regression on the day to take into account the effect of growing on daily feed intake. Then in the next two terms we have A sub 0 and A sub 1. The first one corresponds to the breeding value for level of daily feed intake. This corresponds to the classical breeding value, that it's environmental independent. And A sub 1, it's the breeding value for slope, that is the environmental sensitivity of daily feed intake. We can see as well that P is involved in the model. P is the probability that at a given day there was an unrecorded environmental challenge occurring. And then the following two terms involve the permanent environmental effects, where the first one corresponds to the intercept and the second one to the slope. And finally the residual term. We estimated covariance components using Gibbs sampling and REML, using the software Gibbs F90 and a REML F90. And a model with heterogeneous residual variance was also fitted to assess the sensitivity of the results to how the residual variance was modeled. And basically what we saw is that the parameter estimates did not change considerably. So we were able to estimate breeding values for environmental sensitivity of the genetic potential for daily feed intake on the animals. The level and slope showed to be negatively correlated, minus 0.46 as you can see. And basically what we can say is that a hypothetical selection for increased or decreased daily feed intake would result in increased susceptibility to stress for feed intake. To conclude, we can say that the method is promising and seems viable to identify unrecorded environmental challenge events. It is useful when selecting resilient animals and only productive data are available on a frequent basis. It is general enough to be applied to a wide variety of phenotypic records from different species and useful when dealing with large datasets. Using this method, it is possible to detect different environmental challenges other than meteorological. This is interesting because in most cases there is no information at all of other disturbances other than climatic factors. The inferred probabilities of occurrence of unrecorded environmental challenges proved to be informative and useful to include as a covariate in a reaction norm animal model. And finally, in the ovine example, the negative correlation between the level and the slope shows that a hypothetical selection for increased or decreased daily feed intake would result in increased susceptibility to stress for feed intake. Thank you very much for your attention.